Our project is called 3D Bioprinted Vein-like Heart System.
The problem we face today is that, as of October 2021, 106,834 people who are in need of an organ transplant have already signed up for the transplant waiting list in America. Every 9 minutes, another person is added to this list, and on average, 17 people die each day waiting for organ transplant, as the wait list is estimated to take 6 to 9 months. Of these patients, up to 50,000 of them are heart transplant candidates. Because of the severe organ scarcity, healthcare practitioners face the challenge of life and death. They have to decide who should receive a heart transplant or who should not. Manufacturing a potential heart replacement means that theoretically no one will have to wait 6 to 9 months for the donor transplant list again. We want to save those lives by doing so and combining additive manufacturing technology with stem cell culture to develop a 3D bioprinted heart replacement. It will be manufactured specifically for every patient in need and comprised entirely out of lab-grown human tissue and a pacemaker-like system to control the rhythmic contractions to pump blood.
The three components of our project are the cell culture, the 3D bioprinting, and the electromechanical stimulation. With each component, we believe it is possible to create a vein-like system which can contract. In collaboration with our Department of Biology, we aim to grow living cells into muscle and vascular tissue from stem cells. The stem cells we grow will be monitored daily. To conduct this experiment, we will utilize stem cells placed into 3D printed tissue scaffolds. 3D bioprinting is a type of additive manufacturing that prints live structures layer by layer, mimicking the behavior of natural living systems, using cells and biocompatible materials. Once we have sustainable tissue, we plan to create an electrical system similar to a model of a pacemaker using biocompatible materials such as platinum and titanium.
So what's our business model? Simple. First, we produce the hearts. Next, we sell them directly to the hospitals. From there, they can either store the hearts on their own, or for proper storage, we could store the hearts for them. Finally, we send the hearts back to the hospitals when need be. Our marketing plan is quite simple. We plan to go hospital to hospital selling our product. Why? Because we believe the product sells itself. It saves lives. Next, to get the word out there, we will go to use the outlets of hospital newsletters and local news. People read these things, and it'll just help build our momentum for the product.
There are various competitors out there that are a threat to our device. For example, Duke University has developed a working artificial heart muscle. BioVacor has the ability to contract in the same way as the atria and ventricles of a human heart do. Other competitors are pacemakers. They will continue to be widely used because they have been around for so long and patients trust their usefulness and invasiveness. Traditional operations will continue to compete with our product. For example, coronary artery bypass grafting is an alternative to heart transplantation for individuals with coronary artery disease and left ventricle failure. Moreover, mitral valve surgery can be performed with reasonable survival in patients with severe left ventricle dysfunction and is favored over replacement when possible.
Our team is composed of students from different engineering disciplines. We have 2 bioengineering students, Andrea Colon and Yvonne Fair. Our electrical engineering student is Josh Aras. Peter Gorey is our mechanical engineering student, and our faculty advisor is Dr. Nazar Hagbin.
Obviously, the end goal for us is to be able to produce a batch of hearts ready for the market. However, we must dial back to short-term goals, for this is a little ambitious. Printing a simple 1 to 2 chamber system that works, and then possibly putting a 4-chamber heart system for testing. We must learn to crawl before we walk, so we must start small before we reach the market.
Research and in-depth journal reviews to determine the best and most efficient ways to grow and develop stem cells for 3D bioprinting. We have designed 2 different models which will work in different ways to produce a volumetric flow rate and done some simulations through them. We have also received funding and made purchases of materials to start manufacture our very first prototype. We also plan to complete the project by May 22 and will use the funding raised to present a fully operational model at a conference or 2.